Hello everybody. In this video, we'll look at question number 11 of GATE Biotechnology 2015 paper. So the question is, which one of the following complement proteins is the initiator of the membrane attack complex? Option A, C3A, option B, C3B, option C, C5A, and option D, C5B. So to answer the question, let's understand about complement system and the components involved in it. So the complement system is part of our body's immune system that cleans up damaged cells, helps a body to heal after an injury or an infection, and destroys the pathogens. So the complement system is the frontline defense for our immune system. So it is named as complement system because it was first identified as a heat labile serum component that complemented antibodies in the killing of bacteria. So it consists of serum and cell surface proteins involved in defense against pathogens and tissue damage mediated by antibodies. So it's a major effector of humoral immunity and also plays a major role in both innate and adaptive immunity. So in short, the complement system is a series of events that occur to guide the antibodies to destroy pathogens. The major functions of this complement system are cell lysis, that is, rupturing the cells and killing them, and opsonization, which is a process in the immune system where particles such as pathogens or dead cells are marked for phagocytosis by immune cells, particularly phagocytes like macrophages and neutrophils. So opsonins are molecules that facilitate this process by binding to the surface of the target particle, making it more recognizable for the phagocytes. So another function is chemotaxis, which is release of chemicals or signals that help attract the immune cells. And then activation of inflammatory response by helping recruit immune cells to the site of infection. And finally, immune clearance, which is the elimination of the antigen-antibody complexes. And then talking about what are the components involved in the complement system. So there are over 30 serum and cell surface proteins that are involved. So these are complement components that are inactive in serum but are activated sequentially as a cascade. And complement receptors, which are on the cell surface and recognize activated components, and then regulatory proteins, which are found both in serum and the cell surface and inhibit the activated components. So the components are designated as C1 to C9 or letters, for example, factor D. So these components are mainly synthesized by hepatocytes, that is the liver cells, and they're also produced by blood monocytes, macrophages, and epithelial cells of the gastrointestinal or the genitourinary tract. So the complement proteins are generally activated upon cleavage. For example, the complement protein C4 is cleaved into C4A and C4B. So A is the smaller fragment that's involved in diffusion and signaling, and B is the larger fragment that remains bound to the microbe. The exception is the C2 complement protein, where A is the larger fragment and B is the small fragment. There are three major complement pathways. So they are the classical pathway, the alternative pathway, and the lectin or MBL pathway. So the pathways for complement activation require multiple steps, which are recognition and then enzyme activation and biological activity. So the first two steps, recognition and enzyme activation, they require unique proteins. Let's now dive deeper into the classical pathway. So the classical pathway is part of the adaptive immune system and it relies on antigen-antibody complex to get activated. So the classical pathway is initiated by the antibody binding to the antigen which is the disease-causing pathogen. So the C1 proteins bind to Fc of the antibody. So C1 proteins are C1q, which binds to Fc on the IgM or IgG antibodies to get activated. So the C1q has 18 polypeptides, which consists of 6 arms with globular heads, as shown in this figure. So there are also two C1R and two C1S proteins, which are activated by when bound by the activated C1Q protein. So the antibodies like IgM in generally have two different conformations. So one is the planar structure, which does not have any exposed C1Q binding sites. So this is the planar form of IgM, and it does not have any provision for the C1Q protein to bind to it. And the other conformation is the staple conformation, the staple structure of IgM. So it has at least 3 binding sites for C1Q protein. So as shown is in this figure here. So the pentameric IgM molecule binds to antigens on bacterial surface and adapts the staple structure to enable the binding of C1. So this C1 then binds to a single IgM molecule. And then in case of IgG, so the IgG molecules bind to antigens on the bacterial surface, and C1Q binds to 2 or more IgG molecules. So the classical pathway has now been initiated upon the binding of C1 to the antigens on the microbial surface. So the further steps are discussed in this slide. So the activated C1 proteins, they cleave the C4 proteins to C4A and C4B. So some C4B, the larger C4 component, binds covalently to the microbial surface while C4A remains unbound. So the activated C1 proteins also cleaves the C2 proteins to C2A and C2B. So the C2A protein binds to the surface of C4B, forming the classical C3 convertase complex, which is the C4B2A complex. So this C4B2A binds to C3 protein and cleaves it to C3A and C3B. Now C3B binds covalently to the surface of the microbe. Now C3B here is an opsonin. So opsonins are molecules that bind both to bacteria and phagocytes, which helps in opsonization. So opsonization increases phagocytosis by 1000-fold. So like we just saw, opsonization is just enabling the recognition of the microbes by the phagocytic cells. We now have the C4B, C2A, C3B complex bound to the microbial surface. So this functions as the classical C5 convertase enzyme. So this cleaves the C5 component to the C5A and C5B. The later stages involve inflammation, phagocytosis, and cytolysis by the membrane attack complex. So the C5B, along with other components already bound to the cell membrane, binds sequentially with C6, C7, C8, and multiple molecules of C9 and forms the membrane attack complex. So these C9 molecules polymerize to form a tubular structure known as the membrane spanning channel or the pore. So C5b thus initiates the formation of this complex. So the assembled membrane attack complex is inserted into the lipid bilayer of the target cell's membrane. So this disruption leads to an influx of water and ions into the cell, causing it to swell and eventually burst. So this process is known as cell lysis. Cell lysis results in the destruction of the target cells. The release of cellular contents, including intracellular components and potentially harmful substances, contributes to the elimination of the pathogen or the infected cell. So this is how the membrane attack complex works in the complement system. On the other hand, C3A and C5A bind to the pathogen and recruit neutrophils that destroy the microbe. So the C3B, on the surface of the microbe enables phagocytosis by opsonization. So we have now seen the functions of all the components in the complement pathway, in the classical pathway of the complement system. So now coming back to a question, which one of the following complement proteins is the initiator of the membrane attack complex? So C3A, C3B, C5A, and C5B. So the answer would be C5B, because we saw that it binds with C6, C7, C8, and C9 and inserts itself in the membrane of the cell. So that is the membrane attack complex. So on the other hand, C3A, C3B, and C5A, they help in opsonization, phagocytosis, and help to recruit neutrophils that phagocytose the pathogen. So that answers our question. Thank you.